two types. So we can divide into two parts. Right. So what are those two parts? The first part is called ECF and ICF. Right. So what is the ECF? What is the difference between ECF and ICF? Anyone of you have any idea? ECF, ICF. Okay. So without wasting any time, I'll tell you what is the ECF and ICF. Look, intracellular fluid and extracellular fluid. Intracellular fluid, the fluid part that is found within our cell. So ICF, intracellular fluid, will contain, that is, protoplasm. And the question that have been asked, the ECF is rich in, ICF is rich in. Remember, the protoplasm or intracellular fluid, that is rich in potassium and phosphate. Comparatively, so intracellular fluid is protoplasm only. ECF, extracellular fluid, means the fluid which is found out of the cell, extracellular fluid, right? That is outside of. It is made up of lymph, blood, but comparatively, this is rich in sodium and chloride, right? Note it down, copy it as it is. The blood have 30 to 35% ECF. Blood or ECF is 30 to 30, right? Blood is a part of 30 to 35%. Blood means blood make 30 to 35% part of ECF. An adult person of 70 kilogram, that is a ideal, have 5.5 liter blood with a 7.4 pH value. Note down this part. Then I'll ask you a question. And let's see, do you answer it or not? It's a very common question. I think I have discussed that question. Note down quickly and let me know when you note it down.
Done, sir.
Done. So I have a question. Is there any difference in the pH value of oxygenated and deoxygenated blood? Believe on me, I have discussed with this. Yes, Rabia, you're telling me. Tell me.
Yes, there is a difference.
Very good. So what is the difference? Tell me. Aisha? Or there is no difference? Please note it down. I have discussed you when I was discussing the transportation of carbon dioxide. The pH value of normal blood or standard blood, the pH value of normal or standard blood is 7.4. Is 7.4, but the pH value of deoxygenated blood, but the pH value of deoxygenated blood is 7.35, which is 7.35, which is slightly more acidic. You may get this question in the form of a statement as well, which is slightly more acidic than oxygenated blood. Fine. Plasma. This question have been, by the way, this is very older question and this is a very basic question. You'll hardly get this kind of a question, but nobody can tell that this question will come and this will not come. I have seen a very 20% question are very simple question. Everybody can do that question. And every question, either it's harder, difficult, or it's a easier one. Every question have its 4 marks. So the question was that what is the percentage of the plasma in the blood? So 55% content is of plasma in blood. So out of blood content, 55% plasma, 55% part is plasma. Out of this plasma, I mean the second question, I have already made a star on that. Even the second question I have been asked, out of this 55%, 90 to 95% of plasma, 90 to 95% of plasma is made up of water. What is the rest? 10 to 8%. That is mineral, nutrient, and protein. 6 to 8%, 6 to 8% protein is found in the plasma. I have a question. Whatever we eat as a food, everything is acidic. Everything which seems tasty to you, milk. What is the water? Is it neutral, basic, acidic, slightly basic, slightly acidic? Which we drink? Tell me.
Neutral.
Neutral is that water which is a distilled water you use in chemistry lab, and that it do not have any taste. Neutral is that water which is used in injections. The water which we eat, drink, that is a mineral water. You never purchase. Have you purchased drinking water in the form of distilled water? Every time you go to a shop, you say, just give me mineral water. Correct?
Yes, sir.
That contain mineral, and that is slightly acidic. That's why water have taste. The taste of water change from place to place. Right? Why? Because the composition of the mineral of water changes place to place. That's why the taste of water is changed to place. Right? That is true. After a certain distance in India, every part the composition changes. That's why the water after a certain distance and every part, like even in short distance, the taste of the water is different, right? Because the mineral composition varies largely. So look, the water which we drink, that is also acidic. Milk acidic, fat acidic, sugar acidic, protein acidic, amino acid, right? So everything, whatever you eat from morning to evening is acidic. It get absorbed into your blood and it goes to blood. But when you eat everything acidic, how your blood is basic? So my question is that which component of the blood maintain the basic nature of the blood? Or pH value 7.4. Who will tell me?
Is it plasma, sir?
Is it? It is?
Plasma?
Look. Up to certain extent you are right, but that is not an answer. Actually, the blood plasma have 6 to 8% protein. The name of that protein is albumin, the same protein which is found in egg white, right? It maintains the pH value of blood. It work as a buffer solution. So buffer solution is that solution which do not allow to change the pH value of certain solution, right? So that's why albumin. Note down, please. The blood plasma contain 6 to 8% part. Blood plasma contain 6 to 8% part of protein. It is albumin. It is albumin and globin protein. It is written over here. It is albumin and globin protein. Full stop. Next point. Albumin work as albumin work as buffer solution. It maintain the basic nature of blood, that is 7.4 pH value. Clear?
Yes, sir.
Then there is a lysozyme and properdin, which is also found in the plasma. So lysozyme is an antibacterial. It kills the bacteria and prevent entry of that all insect and all that. Sorry, antigen and all that. And there is one more thing, that is properdin. Properdin is a type of protein. It is always found in the plasma. It will also destroy the bacteria and toxic substance. Please note down. Properdin have been asked. Lysozyme and properdin. Lysozyme and properdin act as antibiotic substance. Properdin have been asked. Act as antibiotic substance. Full stop. They destroy bacteria, viruses, and toxin. They destroy bacteria, viruses, and toxins which enter into blood. Now the next question is very popular. Oh, heparin. What is the role of heparin? Who will tell me? Aisha and Rabia. Heparin. This question has been asked, but it's also very old, multiple times. I don't think even this question will be repeated. Heparin. Even the question I've been asked, from where the heparin is produced. In that question I've been asked. We'll discuss that question in future. From which part the heparin produces? Tell me quickly. Come on. It's a very basic question. So these all are a component of the blood. Plasma, lysozyme, properdin, then heparin. Heparin is a type of hormone. It remains dissolved in our blood. It works as anticoagulant or anti-clotting agent. It do not allow blood to clot inside our body. Right? Why blood do not clot inside our body? Because heparin is anticoagulant. It is found in the blood. When we get injury and blood come out of the body and come out in the direct contact with the air, then heparin stop working. It get destroyed. It stop working and clotting process start. Right? Blood become it changes its form like it first it change like curd form, then it get it becomes solid. Right? Please note down. Heparin is a type of hormone. Heparin is a type of hormone found in our body, found in our blood, not body, found in our blood in dissolved condition. In dissolved condition. It do in dissolved condition. Yeah, sure. Heparin. This is heparin hormone, anticoagulant. It remains dissolved with blood. It do not allow blood to clot inside body. That's why it is called anticoagulant or anti-clotting agent. Anticoagulant or anti-clotting agent. Now, what is the serum? Even this question is very multiple time repeated question. Number of the time it have been repeated, right? So this question have been repeated multiple times, right? So serum, what is serum? Take the blood plasma and remove this heparin or anti-clotting clotting agent, right? This is called serum. Note down. Serum is a blood plasma without clotting agent or clotting factor. Serum is a blood plasma without clotting factor. Serum is a blood plasma without clotting factor. Right. So now this is blood, and normal blood, when we get injury, normal blood clotting takes place in 6 to 7 minutes, maximum 20 minutes, right? If a person have got injury and the blood is not getting clot, blood is coming out, it means he or she have some what to say, some disease or some abnormality, right? Because the normal clotting should take place maximum in 20 minutes, right? This is
Sir, then we
fully,
can see it can be lack of platelets, right?
yes, lack of platelets, that may be diabetic, and there is a one disease called bleeder disease, hemophilia. In hemophilia, blood do not clot, not at all clot. It keep on flowing. Blood clotting process do not take place in hemophilia disease. That is genetical disease. You must have studied this disease in class 12. Have you completed your genetics part? Heredity and variation chapter?
Yes.
It is there. Have you gone through with the hemophilia? Yes, Rabia, Aisha? Hemophilia, have you studied?
Yes, sir.
Yes,
So
sir.
why does blood clotting do not take place in hemophilia? Because there is a clotting factor 8. Sometimes it's a clotting factor 9, which is missing in that blood, right? Due to genetical disease. And this is a type of deletion, right? It's a deletion of gene. It's a gene mutation basically. So it's a deletion of gene. So due to deletion of gene, the formation of clotting factor 8, sometime 9 do not take place. And because of lack of that clotting factor, blood do not clot, right? And the first time this disease had been discovered in the a prince or son of Queen Victoria. His name was Leopold. So Leopold was the son who was affected with disease. He was the first person. That's why this disease is also known as the royal disease. Okay. So look, clotting, when clotting takes place, blood gets separated into two parts: a solid part, which is red color clot-like, and blood plasma. Look, this solid part is made up of RBC, WBC, platelet, thrombin protein, fibrin protein. This is 45%. 55% is blood plasma. It is light yellow in color, made up of hormone, 90 to 92% water, carbohydrate, protein, fat, and mineral. Please note down quickly this one as it is. Please note down quickly this composition. Noted? Done?
Yes.
Fine. So this was blood and left clotting part. Heparin, I have already told you. Blood serum, I have already told you. Yeah, the question. And look, sometime you may get question, but hardly you'll get question from the blood capsules. But let me tell you, some points are important that you need to note it down, because these points are missing in your NCERT. We call them blood capsules. We do not call them blood cells. That we have already discussed that there are some components which are missing over there. So RBC is known as the erythrocyte. It do not have any nucleus and no cell organelle. Right. A very beautiful point I am discussing with you. You know, RBC do not go for aerobic respiration. RBC go for anaerobic respiration. Do you know this fact or not? Every cell perform aerobic respiration in our body. RBC do not. Why? Because if they will perform aerobic respiration, then what will happen? They will consume all oxygen themselves. What they will give to others. Right? So that's why RBC perform anaerobic respiration. RBC is made up of, that is a plasma part and globulin, hemoglobin, right? So every RBC have a component which give red color to the blood, that is called hemoglobin. Hemoglobin is made up of two components, globin protein and iron, that is called heme group. Formation of RBC takes place in red bone marrow. That point must be cleared. Now come to this point. The number of RBC is 4.5 to 5 million per mm cubic. Means 1 ml of blood contain 4.5 to 5 million. 4.5 is for female and 5 is for male. That's why we describe this range. The abnormal rise of, that is more important point for you. These questions, these are the questions. Abnormal rise of RBC, if beyond 5 RBC are there, that is called polycythemia abnormality. Abnormal decrease in the RBC is called erythrocytopenia. Note down both of the points. Abnormal rise is polycythemia, both are disease, and abnormal decrease is called erythrocytopenia. Erythrocytopenia. Size of our RBC may be 7 to 8 micrometer in diameter. The next important point is 100 ml of blood contain 12 to 16 gram of hemoglobin. Formation of RBC is called erythropoiesis. Formation of RBC is called erythropoiesis. Lifespan, 120 days. Graveyard of RBC is called spleen. If somewhere it is written that graveyard of RBC is liver, that is wrong. Graveyard of RBC is spleen. After the death of RBC, means after 120 days, when the RBC dies, that dead RBC goes to spleen. Spleen breakdown over there, right? Spleen is also known as the blood bank, right? Why it is called blood bank? Because extra RBC are collected over here, right? In a spleen. Anemia, deficiency of blood or RBC is called anemia. Clear? All point? And note it down. Polycythemia, erythrocytopenia, erythropoiesis, and graveyard. Aisha and Rabia. Clear? Noted?
Yes, sir.
Come to WBC. This diagram is very, very important. You will get question from it. You will see the question from this point, and every question will be covered by this chart. I have written it. Look, WBC is of basically two types, right? Technically, WBC are known as the leukocyte. They are of two types. agranulocyte and granulocyte. Agranulocyte do not have any granules, they are cytoplasm or protoplasm. Agranulocyte is divided into two categories: lymphocyte and monocyte. Lymphocyte may be of two types: B lymphocyte or T lymphocyte, means B cells or T cells. B cells are produced in liver, right? And they produce antibody. T cells are produced in thymus and they directly attack and kill the antigen, right? Kill the antigen, right? Lymphocyte characteristic, they may be B or T lymphocyte. They are non-motile, non-phagocytic, means neither they move from one place to another, nor they eat bacteria. They produces antibody and destroy the microorganism. This question I've been asked. The lymphocyte cells, basically T lymphocyte cell, they kill tumor. So our body have capability of fighting with the tumor, right? That is tumor means that is cancerous part, tumor, right? Lifespan of lymphocyte is few days to years. Second type of granulocytes are monocytes. They are the largest of WBC. This is the question. They are motile and phagocytic. They engulf bacteria and cell debris, means broken part of cell, right? They live for only 10 to 12 hours. That is the lifespan. Whatever I have underlined, that is the question. Further, granulocytes, the lifespan is 4 to 5 days. They have granule in their protoplasm. They may be of three types: eosinophil, basophil, and neutrophil, right? Eosinophil increase, their number increase in allergy, asthma, and hay fever. This is a question. They help in blood clot dissolving. If suppose that due to internal injury there is a clot of the blood, they dissolve the blood clot in our body. They are non-phagocytic. They are similar like lysosome. They destroy protista, right? Basophils, they release heparin. That is a question which I was asking few minutes before. So heparin is anti-clotting agent. Basophil means a type of WBC which produces heparin, serotonin, and histamine. They are least in count. Even this question I've been asked, which is the numerous WBC. Neutrophils, they are most numerous of WBC. 60 to 65% of all WBC are there, right? They eat harmful germs, and that's why they are phagocytic. So this classification you need to note down. You will get question from this class.
Yes, sir.
Done? Okay. Rabia, done?
Yes, sir.
Next one is blood platelet. So this is not that much important, but their number is 1,50,000 to 3,50,000 per mm cubic, means per ml. Increase is called thrombocytosis. Decrease is called thrombocytopenia. Colorless, they do not have nucleus. Like RBC, they also do not have nucleus, means they are non-nucleated. Lifespan is 1 week. Formation of platelet takes place in karyocyte of bone marrow. That question happened. Megakaryocyte. There is a particular cells. The name of those cells in the called megakaryocytes. Right. So mega means larger. Karyo means nucleus. The cells which have larger nucleus called megakaryocyte. So their formation takes place. Clear?
Yes, sir.
Blood group. Yes. The question comes from blood group. So blood group was discovered by Ladespiner in 1900, and ABO, that is a point to be noted down. A, B, and O blood group discovered by Ladespiner in 1900. After that, AB blood group discovered by DeCastro and Stanley, right? Blood group may be A, B, AB, and O. So name of the blood group is on the basis of their antigen. So do you have idea what is the antigen and antibody? Tell me quick first. Quickly, tell me. Do you have or not? If you don't, I'll tell you. Aisha and Rabia.
Yes, sir.
Fine. Look, anything, what is antigen? Anything which enter inside our body, and when it enter, it trigger our immune system. It trigger our defense system. If it trigger, this is antigen. You put the blood inside the put the water inside the blood. Blood won't react, means body won't react, because blood is not antigen. Put some chemical, body will react. So antigen, any kind of the chemical, any kind of the substance which is not part of body, if it enter inside the body, it trigger the defense mechanism of body. It is antigen. So antigen may be of two types. It may be abiotic or biotic. Abiotic in smoke. If you have allergy with the smoke is antigen for you. Dust, chemical, right? Biological, it may be bacteria, virus, protozoa, fungi, pollen grain, which are not part of our body. When they enter inside the body defense system start working, mean WBC start working. Now, our WBC are the soldier of our body. They kill any virus or any antigen by two ways. First, they are phagocytic type of the WBC. Phagocytic type of the WBC, they eat bacteria and they die themselves. They are suicide bomber like, right? They eat bacteria and they will die themselves. They are called phagocytic WBC. Non-phagocytic, what they will do, they will not eat bacteria or virus. When bacteria and virus will come to them, they will release a type of chemical. That chemical is a type of protein. It gets dissolved in the plasma. When this chemical will come in contact with bacteria, bacteria will die. But they will not die. They are not suicide bomber. So they are this protein which is released by this WBC, is non-phagocytic. This is called antibody. Now is it clear? Antibody and antigen. So antibody is a type of protein that remains dissolved in our blood. Is that clear? Antigen, antibody.
Yes, sir.
So every RBC have antigen in their body. A blood group have A antigen and B antibody. So the name of blood group is basis of antigen. B blood group, B antigen and A antibody. AB blood group, that is important. It have AB antigen but no antibody. That's why it is universal recipient. It takes blood from any blood group. AB. O blood group, it do not have antigen, but it have antibody. That's why AB antibody. That's why this is the universal donor. Right? Clear?
Yes, sir.
What is RH factor? Look, RH factor, I think you must have studied in genetics, heredity and variation. Did or not? RH factor, positive and negative blood group. This is there. ABO blood group is there. Blood group is there. Multiple allelism. Have you gone through with that or not? No. Have you done genetics in 12th class?
Yes, sir.
Rabia? So, have you done ABO blood group? It is there in your NCERT as well.
Yes, sir.
That is called multiple allelism. So, what is A positive and B positive? Actually, RBC may or may not have a protein cover around it. That is called RH protein or RH factor. If RH factor is present around RBC, we will call positive blood group. If RH factor is absent, this is called negative blood group. Clear? Why we call it RH? Because this has been discovered in rhesus monkey and discovered by Ladistiner and Weiner. Note down who discovered RH factor. If you give a wrong blood group to person, due to wrong blood group, what will happen? If A blood group is given to B or B is given to A, what will happen? RBC will clump, they will stuck, and this process called agglutinization. RBC will clump, agglutinization will take place, and a person will die. Remember the term agglutinization. What is agglutinization? Is that done?
Yes, sir.
Okay, we are running short with the time, so today we have covered a very small amount of very few chapters, few part of this chapter. So next class we'll start from here onward. I will discuss with you HDN. Even HDN is there in 12th class syllabus. HDN. So we'll start from here onward. Have any question?
No, sir.
So next class we'll start from blood onward. When we'll complete the blood, before starting the circulation, we'll do some question. Then we will start circulation. Fine. So bye-bye. Good night. Take care. Bye. Rabia.